All right, so welcome back, grade 12s. Last class, you can flip over the slide. So last class, we talked about DNA, and we talked about some of DNA's replication. So we have a little bit of a fun meme, and I don't know if anyone's familiar with Nacho Libre, but it's been requested by a student, so we have a little picture for you to tee it up. All right, so next slide. So some of the objectives for today. We're going to review the central dogma of replication. We're going to talk about the semi-conservative model again, just to make sure that that's clear, because that's going to tee us up for what we're going to go through later. What we're going to do later is talk about technologies that are involved with DNA. Talk about some different types of mutations, and we're going to talk about some ethics, okay? So that's kind of where we're going to be heading today. So I want you to keep in mind a guiding question when we're going along today. This is the guiding question. So how has the knowledge and molecular nature of genes and DNA led to new biotechnologies and treatments of genetic disorders? So just, I don't need an answer right now, but just kind of keep that in the back of your mind while we go through today's lesson. So, central dogma. Who here remembers the central dogma? Oh, we got one keener who read the readings over the weekend. So, Stephanie, what do you remember about the central dogma?
That's where you talk about the process that goes from the DNA to proteins.
Right. So, DNA goes to proteins. Is there a step in the middle there?
RNA.
RNA, right. So DNA goes to RNA. RNA gets encoded into proteins. Can we go the other way? Can proteins turn into RNA, RNA turn to DNA? For the most part, no. So for the scope of this class, let's say no. All right. There are some cases, but that's far extending past beyond what we need to cover in this class. Okay. Okay. Next slide. DNA replication. This was kind of where we finished off last class, right? Sort of. So how do you remember the steps in DNA replication? What's the first step? What's the first thing that happens? Yeah, Taylor.
The DNA has to unzip.
The DNA has to unzip. So there's a certain enzyme that unzips it. Do you remember what the enzyme was called?
I think it's helicase.
Helicase. Very good. If you didn't remember, it was right there. All right. So once the DNA is unzipped, what's the next step? That we're unzipped. All right. So we have two open. We have a 5 and a 3 that are open. All right. Do we just leave that open, or does something else happen?
More enzymes.
More enzymes? Actually, what happens is proteins are binding to these exposed ends of the 5 and the 3. That way we stay unzipped so that we can replicate. All right? Because if not, they just want to snap back together, right? Because that's kind of what it's used to. All right. So we have the helicase. It unzips our DNA. We have the proteins that bind to the DNA, keeping it open. Then what happens? If you can't remember, there's a nice, helpful cheating guide right up here.
It gets copied.
It gets copied, right. So we talked about 5' and 3' on the ends of the DNA. What direction does DNA copy?
5'.
5'. 5' to 3'? What are we seeing here? What does that say? 3 going to 5. Okay, so it always copies 3 to 5, small to big. There's an enzyme that's involved in copying DNA. What's the enzyme?
DNA polymerase.
Polymerase, right. And we talked about 2 different types of polymerases. We're going to talk about that a little bit more today, all right? But DNA polymerase is involved in the copying of the DNA, the transcription of the DNA, all right? So DNA is considered semi-conservative or conservative or dispersed. Do we know?
Semi-conservative.
Why do you say that?
Because it doesn't create just new strands. There's a new strand and an old strand in the
Right.
DNA.
And actually they did, that's a perfect answer, Stephanie. And Stephanie's over there, Taylor. That's a perfect answer. And they actually showed this experimentally. What they did was they'd take a parent DNA strand and they'd inject it with a heavy radioactive isotope. So it would bind to the old DNA. And as the DNA copied itself, they could see that there was one strand that was heavier than the other. So that proved that DNA is semi-conservative, all right? That's part of the central dogma of the DNA. Okay, so enzymes. We talked a little bit about enzymes. Helicase, it unzips the DNA, right? And then once the DNA is unzipped by the enzyme, we have some proteins that are binding to the open ends of the DNA. Those proteins, which have they're held together by hydrogen bonds, remember, right? So those proteins are blocking those hydrogen bonds from fusing the DNA strands back together. DNA polymerase number 3. I can't remember who said polymerase is involved in transcription, But polymerase number 3 works on the leading strand. Remember what the leading strand is in our replication model. There's two. There's a leading and a lagging. Does one sound faster than slower? So which one do you think is faster here? Which one looks faster? This is a leading strand, and this is a lagging strand.
The leading strand.
The leading strand, because it's synthesizing in one direction seamlessly. All right. Lagging strand goes the other way, so it's still going 3' to 5', because remember DNA runs anti-parallel, but it's working against the genome. So we have these little chunks that are made. So that's a different polymerase. The DNA polymerase 3 only works on the leading strand, and it's faster. Next slide.
Sorry to interrupt. Everything good?
Yep.
Yeah. Okay, I'm in my office at 3:30 if there's any emergencies, okay? And did you guys all get your feedbacks? Your feedback? Okay. All right, good
All
luck.
right. So now we're on to the lagging strand, the slower strand. Thanks, Darren. So DNA polymerase 1 is what acts on the lagging strand. So 3 leading, 1 lagging. All right. So keep that in mind. I know there's a lot of polymerases going on. It's going to get worse. Trust me. So try and keep that in mind. Make a note of it. All right. It still works in the same direction, but it's slower because it has to work in small segments because it's working against the direction of replication. So it's got to work backwards almost. All right. So it gets these little chunks, and it doesn't put them all together perfectly. So these little chunks with gaps. To fill those gaps, because we can't have gaps in our genome or else we got problems, we have another enzyme, ligase. So ligase is kind of like glue. It glues all these short fragments together and makes them so they're seamless so they can be copied again later on or read. Next slide. So we know a lot about how DNA is replicated. And with our knowledge, there's some applications that we can use in technology. One of them is the Human Genome Project. So everyone here has some technology I can see. Could you all quickly Google the Human Genome Project? Just a quick Google search. I'm going to hand out some paper while you do that. When you get the paper, just look at the first few questions and see if you can answer them just through reading what you're seeing on one of the first things that come up on the Human Genome Project. If you get an answer, just shout it out, because I don't have a copy of it in front of me. You may go.
You want us to answer these questions?
Yeah, just out loud. So if you find one, just let me know what question you're answering. So, yeah, Taylor, question 1. Question
The
1.
Human Genome Project began in 1990,
1990. Does it say when it finished?
2003.
1990, 2003. How many years is that?
13.
Whoa. So that's a fairly large project. Okay. What's the next question?
How many genes do human beings
Yeah.
have?
That could be useful. How many genes do we have on average?
19,000 to 20,000?
19. That seems like it might be a little low.
That's just protein coding
Protein.
genes.
Oh, yes.
So remember our
2,300.
that also seems a little low.
100,000.
100,000 is more. We're getting a little closer, okay? So remember, we're starting out with DNA, and we're coming down to RNA, and then we're getting the proteins after. So those proteins might be what you're getting.
Protein coding
Yeah.
genes.
There you go. Okay. So, sorry, 100,000? So it's a factor in the hundreds of thousands. That's a lot of genes. Do they all, what's the next question? It has to do with junk, right?
Yeah.
Yeah. Are they important to us? Do they all encode stuff? What is junk? What is this junk DNA that I'm talking about? Yeah.
Junk DNA is DNA that doesn't code for protein.
Yeah, it's just there. It's kind of just filler, right? So we have 100,000 genes. Not every one of those is going to encode for something awesome. Some of it's just kind of there, all right? Whether it's through evolution or just, it's probably just evolutionary baggage that was useful at some point in the past, but not so much right now. All right, how about the next question? What do we got for the next question?
Is it fully sequenced?
Is it fully sequenced? So, is it fully sequenced? Is it finished?
Yeah.
Yeah. So we have sequenced the whole thing. And then I think it says something about, have we sequenced any other genomes? I believe I put that.
Yeah.
Yeah. So does anyone know if we have, or if we have, what kind of examples would they be? Yeah.
Fruit fly?
Fruit fly, Drosophila. That is, as you go on in science, you're going to become very familiar with the Drosophila. Okay. Any other ones?
Dogs.
Dogs? I think so, but let's think science. What do we do science experiments on? Sometimes
Monkeys.
it's, what's that?
Monkeys.
Monkeys? Yeah.
Worms?
Worms? What about mice? Lab rats? Right? Those two for sure have been sequenced. I think you're right, David, but I can't say for sure. So I don't want to perpetuate some misconceptions if I can't say for sure. So I can get back to you on that one. But yeah, Drosophila, lab mice. Two very common experimental agents in science, bio, that kind of stuff. Make sure you, whether or not that's ethical, I think we're going to have to talk about it a little bit later. Okay? I think we got 2 more questions.
How much was it anticipated the project would cost?
Yeah. Does it say how much it cost? How much they thought it would cost? How much it ended up costing?
Costed $3 billion.
$3 billion with a B. Yeah. And I think they anticipated it to be $3 billion, and I believe it cost $2.7 billion. All right. So that's some pretty big capital that we're putting into sequencing a human genome. So do you think that's a worthwhile investment? Because we're at a point right now where you can't just dedicate billions of dollars to certain frivolous things. What do you think? Is it important that we know our genome? Yes, no.
I don't
Yeah.
think we need to know.
You don't
it.
think we need to? Right. That's kind of in the eye of the beholder. I would argue that it's important to, and we're gonna get into why knowing genomes might be important going on. Last question, I believe, is is it dangerous that we know genomes? Could something bad come out of that?
Could be,
Could
depending
be.
on who uses it. And what
Yeah,
they use
like-
it for.
It could possibly be used for bad. So these are the things that I want you thinking about. Remember that focusing question that we talked about, that there's a lot of things at play here when we're discovering new biotechnologies. Okay, so there's a lot of factors that you have to consider as you're progressing through research like this. Last question. Most things cost money or can be transferred into the economy. Is there any businesses that you can think of off of the top of your head that might profit from DNA sequencing? I saw one actually on the TV last night.
Probably like pharmaceutical companies.
Pharmaceuticals could definitely profit from that. But what about a service? Is there a service that somebody says, give me your DNA and I'll give you a service? Have you seen a commercial for that?
Like screening for.
Screening? Yeah, that'd be yeah. But what Ancestry DNA. So what Ancestry does is you give them a little swab of your spit, they send it off, and they check for DNA markers that are from different land or from different peoples of different places. All right, it helps them figure it out. But you're giving them a piece of you. You're giving them your A, C, T's, and G's. And it's kind of muddy like that. Another one that I thought of was 23andMe, where it just sequences your genome and then sends it back to you and says, this is what you got. But what do they do with your sample after that? I don't know. I don't know if they tell you. So that's about all I have for the human genome.
Would you get your sequence?
Would I get mine sequenced? I think I would. I think it'd be cool. But that's up to you.
How much does it cost?
Good question. I think it's $250. So remember, $2.7 billion, we're all the way down to $250. All right? So that's kind of how efficient we've got at sequencing genomes. And we've turned it into a private industry, which I think is kind of cool. Okay, so we're going to talk about some DNA technologies quickly here. Recombinant DNA. So when we start fiddling around with DNA, sometimes we slice it and put it back together. So recombinant DNA is DNA made from 2 different sources of the DNA. So it creates a genetic transformation, as I've outlined here, where the cell or the DNA or the living organism has different DNA sequencing in their cells now because we've done this process. It expresses the new DNA, and recombinant DNA is involved in several methods of biotechnology. So, restriction endonucleases. When we see ACEs, what do we think? Enzymes, right. ACEs means enzymes. Okay. So, there are a lot of restriction enzymes. We call them molecular scissors. So we send them in. We've actually derived them from a lot of microorganisms. So the most common one we use is from E. coli, actually. It's called EcoRL, I believe. And what they do is they come in, they cut into the DNA, and they break the double bond, and they get in, and they break it apart. So they've cut the DNA, 2 ends. Those 2 ends, depending on what we're using, sometimes have this little nub sticking out, called those sticky ends. If you have one with a sticky end, it's very useful to insert new DNA because it has a massive bonding potential with these open ends. So that's how we induce new DNA. Sometimes the different the different molecular scissors, as you will introduce ones that split right down the middle. Maybe it stops without those sticky ends coming out. Those are called blunt ends. You need to know that those are possible as well, but they don't have as much application for biotechnology. So I've just included that, but it's not as important as those sticky ends. So yeah, here is the EcoRL that I was talking about. So what do? Comes in, sees the double DNA, comes in and it cuts, because they're molecular scissors, cuts them open, and see how we have all of this bonding potential here. We have these sticky ends, as you were. That's where the DNA that we introduced would bind to, and then it becomes part of that organism's DNA. So when we're looking at the DNA, PCR allows us to see synthesize large amounts of DNA from a tiny little bit. So say we have a tiny little bit of DNA and we want to do a bunch of studies on it. Drosophila, for example. So we take a little piece of the Drosophila and we put it in vials. We boil it, or we heat it up really hot so those hydrogen bonds break apart. As it cools, they stay broken apart, and we add our favorite enzyme, polymerase. So this one's called Taq polymerase. So now we have DNA polymerase 1 and 3, and Taq polymerase. So make sure we got all these straight for our test. So we're gonna add those together, and they synthesize off of the DNA. It helps us create more DNA because it uses the parent ones that have been broken from the heat, and it synthesizes new ones. So yeah, just the procedure, you heat it, you cool it, you add the Taq polymerase, and then you repeat it several times to get the amount of DNA that you want. So PCR generally is a step before gel electrophoresis. I have a little video here on gel electrophoresis. Can you skip the ad?
In this video, I will be discussing how gel electrophoresis is used to separate DNA fragments of different sizes. So let's say you have a sample that has a bunch of copies of this piece of DNA, and you use a restriction enzyme to digest this molecule.
Like that EcoRL, for example.
Here is the restriction site, so we cut here and create a small fragment of DNA and a larger fragment of DNA. Now I want to be able to isolate this fragment to use later in my lab. Right now, I just have a sample containing some medium-sized pieces mixed in with some smaller-sized pieces of DNA. I can use gel electrophoresis to separate all of these medium-sized molecules from all of these small molecules. How do I do that? So this procedure uses a gel, usually made of agarose, that is placed in a buffer that has ions that can carry current. The gel contains indentations called wells, where we inject the DNA sample that we want to separate. We then apply an electric field that has a negative pole on the side of the gel with the wells and a positive pole on the other side of the gel. We should remember that DNA is negatively charged due to the phosphate groups in its backbone. When the electric field is applied, the current is running through the buffer. The negatively charged DNA fragments move away from the negative pole toward the positive pole through the agarose gel. In this process, the larger the DNA fragment, the slower it moves, causing the fragments to separate based on their size as they travel. In this case, let's run 2 samples through our gel. In the first well, let's run an undigested sample that only contains the full large segment of DNA that has not been cut by the restriction enzyme. In the second well, let's run the sample that has been digested by the restriction enzyme, resulting in this medium-sized fragment and a smaller fragment. As the gel runs, the smaller fragment will move faster through the gel than the larger fragment, and we end up with something that looks like this. In the first lane, we can see that our large undigested molecules do not move very far in the gel. In the second lane, we can clearly see two separate bands of DNA. We have successfully separated these two different sized fragments, the medium here and the small here. And we can cut either one of these fragments out of the gel to use the DNA in further lab protocols, perhaps by inserting it into a plasmid and transforming it.
Okay, so that's about it for that. So you saw how they mentioned that you could use the restriction enzymes. It would cut the DNA, you get the different sizes, all right? So say we're trying to get to that smaller size, and we're gonna use it to induce DNA into something. We could find out, based on its size, that we were getting the desired DNA that we needed. And then I think it mentioned something about plasmids at the end. So let's talk about plasmids now. All right, so plasmids, does anyone know what plasmids are? Do you remember? Kinda. So these double-stranded DNA circular things that exist in bacteria. All right, so they're just in the cytoplasm of bacteria. They're very good in biotechnology for inducing or acting as a vector, an agent that gets you from one place to another. Think physics. Gets you into the DNA of the bacterial cell. All right. So what we're doing is we treat the plasmid with those restriction enzymes that we talked about. There are several, but let's just use EcoRL for example. So we cut into the DNA, we create this little gap with those sticky ends. We would run PCR and gel electrophoresis, get that small strand, for example, it could be the larger one, but let's say the small for this case, and we insert it back to the vector or the plasmid with those sticky ends. It'll bind, and to help that binding, we're going to put some ligase in there too. Remember ligase from earlier? That's the glue. So we're going to put that in, and we think we have created a new DNA for the bacteria, but we can't be sure. Why could we not be sure? How big are bacteria? Not big. Can we see their DNA, even on the microscope? Can we usually see DNA on living things? Not that I know of, right? Okay, so what they do is they put a little bit of an antibiotic resistant sequence in that gene, and then they expose the bacteria to those antibiotics. And if they live, they know that it took, and that the plasmid has been uptook with the DNA that we've added to it. So next. So last slide for me. We're going to talk about, so that DNA that's taken in, it's created a new DNA sequence, all right? It's changed how it is because you have inserted something. So I want you to think of, what's the word that you think of when your DNA is changed? Starts with an M.
Mutation.
Mutation. All right.